Hello everyone and welcome back to our genetics lecture series. In today's session, I would like to talk about gene organization and chromosomes. Before doing this, I would like to make few announcements, and the first one is on our first exam preparation session focused on genetics topics. And those face-to-face sessions are going to be on the 19th and 20th October. So those sessions are mandatory, so we expect all of you to turn up. And the purpose of all of those exam preparation sessions is to address potential topic-specific questions, and we will also be solving mock MCQs, short answer questions, and potentially some long answer questions to prepare you for the end of the year exam in summer term. So if you have any questions on the lecture content, and this exam preparation session is going to be dedicated to genetics, please use the dedicated Padlet on Blackboard. If you go on Blackboard, then you have to click on the link, exam preparation sessions, and this will lead you to our Padlet. And there is already one question. Well done. But it would be brilliant if you could ask as many as you want to make most of the sessions. If you do like to post a question, please do it until the 15th of October to allow me prepare some slides to address this. Good. So before talking about gene organizations and chromosomes, let us recapitulate what we have learned about translation and protein biosynthesis. We have learned that the central dogma of molecular biology states that information passes from DNA to RNA and subsequently to proteins. However, proteins cannot pass information back to DNA. So this is a one-way process. We have the DNA, which gets transcribed into mRNA, and finally this mRNA gets translated into proteins on ribosomes. And this is a one-way process which cannot be reverted. Translation involves tRNAs, mRNAs, ribosomes, and specific enzymes. In simple terms, all proteins are polymers of 20 different amino acids, but there are only 4 different nucleotides in our DNA. The protein coding information is read by the cells in blocks of 3 nucleotides, sometimes referred to as codons, and each of those codons specifies a different amino acids. There is also a set of very defined rules that specifies which nucleic acid codon corresponds to which amino acid, and this is our genetic code. In the genetic code, codons are made of 3 bases, and this code specifies an amino acid. With 3 bases, we have 64 possible permutations. With 3 codons corresponding to stop codons, this leaves us 61 combinations that can code for specific amino acids. Therefore, many amino acids have more than just one tRNA to which they can attach, and this is something we have learned in our last session. We have also practiced to use the genetic code table and also the genetic code wheel. So what are the genetic code characteristics that we need to remember? First of all, we need to remember that genetic code is specific. We also need to remember that the genetic code is universal. The same code is used in all living organisms, in prokaryotes, but also in eukaryotes, including humans. Moreover, genetic code is degenerated. That means, although each codon corresponds to a single amino acid, one amino acid may have more than one codon. And as we have seen in the last session, arginine, for example, has 6 of those. We have also learned in our previous session how translation actually looks like. So in the first step, small subunit of the ribosomes is bound to mRNA. This allows tRNAs to enter the complex, and the first one is tRNA, which corresponds to the start codon, AUG. Once this happened, the large subunit of ribosomes can enter the complex, and now translation can start. And this step is referred to as initiation of translation. During the formation of the complex, we have mRNA present in the complex, small and large subunit of the ribosomes, and there are also tRNAs present. During elongation, the peptide polypeptide chain grows slowly, and this is a result of new tRNAs entering the complex. The already used tRNA is released, and this allows the next tRNA to enter. During the termination, a so-called release factor enters the complex, and the release factor is a protein. This leads to a dissociation of the complex, and we end up with new 3 small subunit of the ribosomes, 1 large subunit, mRNA, and our newly formed polypeptide chain. In today's lecture, we will look at the organization of the DNA into chromosomes. And the learning outcomes are as follows. After watching the videos for this lecture and after revision, students should appreciate the necessity for efficient packaging of DNA in human cells. They should also be able to explain the terms nucleosome, solenoid, and chromatin. They should understand the difference between euchromatin and heterochromatin and know the major chromosomal landmarks, including telomeres, centromeres, and replication origin. Moreover, you should be able to recognize normal and pathological karyograms and understand the impact of telomere shortening on aging. If we look at viral and bacterial genomes, at the genomes of rather small organisms, then we will recognize that viral and bacterial genomes are relatively small. And the size of those genomes is usually also related to the nucleic acid present in those organisms. And in viruses and eukaryotes, those nucleic acids are largely devoid of proteins. So in those organisms, we have usually a single nucleic acid molecule, either DNA or sometimes RNA in viruses. And as I mentioned before, they are largely devoid of associated proteins. And again, they are much, much smaller than eukaryotic genomes. For a simian virus 40, the size of the genetic information for the simian virus, which is 45 nanometer, the DNA is 1.4 micrometer. So even here in those quite simple and small organisms, DNA needs to be packaged because again, micrometers, one micrometer contains 1000 nanometers. In a phage, which is slightly larger type of virus which infects bacteria, with a lambda phage having a size of 58 nanometers, the size of the genetic material is 16 micrometers. Again, this actually makes packaging very, very necessary. In coli, which are obviously bacteria, the need of packaging becomes even more evident. Those bacteria are actually quite small, with a size of approximately 0.5 times 2 micrometer. However, DNA in E. coli is 1200 micrometer long. So what is the difference between bacterial and eukaryotic or human genomes? And this is something we already discussed in one of our previous lectures. The first obvious difference is obviously size. Both bacterial genomes are also very efficient. because coding sequences occupy 90% of the genome. In contrast, in humans, the protein coding genes occupy only 1.5% of the DNA. Also, human genes range in size from several hundred base pairs to 1 million base pairs, with an average gene being 27,000 base pair long. And this is something we understand nowadays quite well, because in the 90s, the human genome has been sequenced for the very first time, and it has been largely recognized that the spacing between pairs for human genome is around 3.4 Armstrong, with 3.2 billion base pairs in total. So the total length of the human genome, including coding and non-coding sequences, is round about 2.2 meters for every single cell in our body. So 2.2 meters, this corresponds to quite a lot of micrometers. However, the human nucleus is pretty small, with merely 11 micrometers on average. And what you can see here is actually a microscopic image from an experiment conducted in my lab. And what you can see here are the nuclei stained in green. This is a scale bar of 10 nanometers, and I hope that you agree that those nuclei are indeed round about 11 micrometers in diameter. This actually generates a huge problem, and in order to highlight this problem, I would like to use an analogy. Fitting in such a long molecule of DNA into such a small nucleus is similar to trying to fit a rope which is 220 kilometers long into a shopping cart with a size of 1.1 meters. And 220 kilometers is round about the distance from Reading to Calais in France. So again, we certainly do have a packing problem. Therefore, packaging and access to genetic information in our cells must be regulated. And the solution here are structures called histones. So what are histones? Histones are proteins, and there are quite few of those. We have the core histones, H2A, H2B, H3 and H4, and also a linker histone called H1. Those histones consist of 25% arginine or lysine residues, and those are both basic amino acids. And this is quite important because the charge of those amino acids allows them to bind to the DNA, which has a different charge. Histones form octamers. That means that each histone unit contains 8 histones. So what you can see here is one nucleosome bit, and we have the linker DNA. And the linker DNA is actually the region in the DNA where our H1 linker histone binds to. What you can see here on this image is electron microscopic image of DNA bound to histones, and we are calling this structure 10 nanometer fibers, which consist of nucleosomes. This is another very, very nice picture generated by electron microscopy showing the linker DNA regions and the nucleosomes shown here. So again, a nucleosome is a complex of 8 histones and DNA. Between each nucleosome unit, we have naked linker DNA, which then can be bound by H1 linker histone. Obviously, by doing this, we still have pretty long and quite linear DNA. So we still have to increase the level of compaction. And the next level here is called solenoid. Solenoid is in simple terms a supercoiled arrangement of DNA in eukaryotic nuclear chromosomes produced by coiling continuous string of nucleosomes into a structure. And the solenoid defines the packing of DNA as the so-called 30 nanometer fibers of chromatin and results from helical winding of nucleosome strands. Again, here we have our histone octamer forming the nucleosome. If those structures are supercoiled, we are referring to them as solenoid. And this is quite obvious. This small nucleosome unit is 10 nanometer small, and solenoid is slightly larger with 30 nanometers and contains several histone octamers, DNA, and the linker histone in the core. So let's have a quick look at the 30 nanometer fiber using again electron microscopy pictures. As you can see here, we reach another level of compaction by forming those solenoids, and this is actually quite efficient already. However, this is not the end of the story, because nucleosome formation compacts DNA sevenfold. However, a helical stack of those solenoids provides up to 10 to the power of 4-fold condensation. So we have much more compaction here. Now our DNA finally resembles packets, and we are referring to those packets as chromosomes. Chromosomes are most prominent in duplicated form during cell division, during the so-called mitotic metaphase. But we are going to be looking at mitosis and meiosis and in a dedicated lecture in the future. So, what are the orders of chromatin structure you need to remember? The first is obviously naked DNA without any proteins, and this is quite long molecule. Then we have the nucleosomes formed by histone octamers, DNA, and the linker histone between each of those nucleosomes, and this is referred to as the 10 nanometer fiber. Next level of compaction is solenoid, which is a stack of nucleosomes, and then we have the chromatin fiber. Lastly, we have chromosomes, and those are the level of compaction you need to remember. To close this session, I would like to show you a brief video revising what we have learned so far.
In this animation, we'll see the remarkable way our DNA is tightly packed up to fit into the nucleus of every cell. The process starts with assembly of a nucleosome, which is formed when 8 separate histone protein subunits attach to the DNA molecule. The combined tight loop of DNA and protein is the nucleosome. Multiple nucleosomes are coiled together, and these then stack on top of each other. The end result is a fiber of packed nucleosomes known as chromatin. This fiber, which at this point is condensed to a thickness of 30 nanometers, is then looped and further packaged using other proteins which are not shown here. This remarkable multiple folding allows 6 feet of DNA to fit into the nucleus of each cell in our body, an object so small that 10,000 nuclei could fit on the tip of a needle. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope, chromosomes.
With this, I would like to end the session, and I'll see you in session number 2. Thank you very much.